In this lecture, we're going to discuss the labeling techniques that are used in immunoassay. So prior to this, we've talked about antibodies and antigens and their interactions. Today, we're going to learn about how those are used together in laboratory testing. We're going to start with chemiluminescence. We talked about chemiluminescence a little bit in clinical chemistry. So we talked about how a chemical reaction, usually with an enzyme, creates a color. Well, this is going to be somewhat similar, but the luminescence is going to come from labels that are attached to an antigen or an antibody. We're going to use this process to detect different proteins, viruses, nucleotides, and nucleic acid sequences. There's two different ways we can do this: competitive or sandwich immunoassays. The first one, here's the definition, then we'll see a picture. But you do need to memorize the definition or be able to recognize it. A competitive chemiluminescence is the fixed amount of labeled antigen competing with an unlabeled antigen from a patient specimen for a limited number of antibody binding sites. The amount of light that's emitted from this is inversely proportional to the amount of antigen measured. So let's look at a picture. So here you can see we have the antigen in the serum sample. Okay. We're going to mix it with an antigen with a chemiluminescent label stuck to it. You see this little star splash here. We're going to mix these together in a test tube with antibody. They're going to compete for antibody binding sites. Okay, you can see both antigens fit. And you're going to see they're going to create a binding that's going on here. So as you can see by our definition on the top, the amount of light is inversely proportional to the amount of antigen measured. Okay, so the antigen in the serum sample of the patient took up the majority of the binding sites. Therefore, there wasn't much left for the other antigen to bind, so there was less light or less color produced. Now the sandwich has an additional step to this. This is where a sample antigen binds to an antibody fixed to a solid phase. The second antibody is labeled with the chemiluminescent label, binds to that antigen-antibody complex, and the emitted light is directly proportional. So where the other one was indirect, this one's going to be direct. Here's how the sandwich works. The solid phase is usually adheres to the side of a test tube. We add the antigen, which is in the patient's sample. So we take the patient's serum, add it, and any antigen that's there is going to bind to the antibody that's attached to the wall of the test tube. We're then going to add a second antibody with a chemiluminescent label. So you can see these fit very well as well over here on the side. So we get the antibody attached to the wall, the antigen, and then the antibody with the chemiluminescent label on top. The more binding that occurs in this process, the more light or color reaction is going to occur. So that's a sandwich. You can see it does look like a little sandwich. Okay. All right, so we do have some of those labels. You saw those little antibodies that had, or antigens that had the little labels attached to them. What they usually use is a luminol, acridium ester, something like that. And the enzymes that are used for the indirect labels are usually alkaline phosphatase, horseradish peroxidase, or beta-galactosidase. You are going to have to be familiar for memorizing a couple of these. I always think horseradish peroxidase is really easy because people like to put horseradish on their sandwich. Okay, so that helps you kind of associate that one. Alkaline phosphatase we talked about in clinical chemistry, so that one's easier one to memorize. Beta-galactosidase is a little bit tougher because that's not really a word that you've seen, but you do need to be able to recognize these as chemiluminescent labels. Next is the enzyme immunoassay. This is a measurement that provides actual numeric results, and in order to be used in an enzyme immunoassay, it has to be very stable, very specific. It has to have absence from the antibody or antigen, and there can't be any alteration by an inhibitor within the system. Here's how this procedure works, and we're actually going to perform this later on in the quarter. A plastic beater plate is coated with an antigen that reacts with antibody in the patient's serum. The beater plate is then incubated with an enzyme-labeled antibody, and if the antibody is present, the enzyme-labeled conjugate will react with the complex on the bead. Enzyme activity is then measured. Okay, so we're going to do like a HIV simulator test later on for that, and it actually uses the beads. It's kind of neat. So here are some of the types that we see with enzyme immunoassay. You're going to see this refer to enzyme-linked immunosorbent assay, which is essentially works the same way. So we're going to use the same pictures that it's pretty interchangeable in how they work. So the first one's direct. You can see we have an antigen that combined to an antibody with a, a tag on it. Indirect, we have an antigen, then we have an antibody, and then we have a secondary antibody. The capture antibody has an antibody with an antigen, then another antibody, and an antibody. And the competitive is, again, they're going to compete for binding sites. So here's an example of how this works in the test tube for a non-competitive enzyme immunoassay. I'm going to give you a little hint. This may be a test question. Okay. So the first thing that we do is we use a test tube or a plastic cuvette, and it's going to have the solid phase antigen attached to the outside. We're going to add the patient sample, who's going to contain the antibody. Let's pretend we're doing, I don't know, let's hepatitis B antibodies. Okay. So in the patient sample, if they do have antibodies to hepatitis B, you can see those antibodies are going to bind with those antigens on the side of the test tube. Okay. We're going to do a wash step to get rid of any excess antibody that could possibly be floating around. Then we're going to add an enzyme labeled anti-immunoglobulin that will react with any antibody that's left attached to that antigen. This is going to create a antigen, antibody type of scenario. And when we add a substrate, it is going to react with the label on that secondary antibody. The color development is directly proportional to the amount of patient antibody present. Okay, so that's how non-competitive enzyme immunoassay works. Next, we're going to look at some immunofluorescent techniques. This is the labeling of an antibody with a fluorescent compound that has an affinity for proteins to form a complex. That complex can act with an antibody-specific antigen, and it's very highly specific and sensitive. I remember doing these in the first hospital that I worked at for flu testing. We would actually have a slide. We'd put the patient's nasal smear, from the nasal swab, we'd rub that on a slide, and we would overlay it with a fluorescent antibody. And if the flu was present, it would attach to that flu, and the conjugate would create a fluorescent green color. So let's look at a little bit about how that works. So for the direct immunofluorescent assay, it's where there's a conjugated antibody used to detect an antigen-antibody reaction. So this one refers to syphilis that we see here. So here we have the actual syphilis antigen stuck to a slide on the very, very bottom. Then we have the treponemal antigen to another specificity added to this. We have a conjugated antibody, a fluorescein isothiocyanate, and then an antibody to a treponemal antigen. So we've got a couple different layers that we're adding to try and create that color, or fluorescence, I should say, because it is fluorescent. We can have an inhibition immunofluorescent assay. This is a blocking test in which an antigen is first exposed to the unlabeled antibody, then to a labeled antibody, and finally washed and examined. If the unlabeled and labeled antibodies are both homologous to the antigen, then there's no fluorescence. Next is the indirect fluorescent assay. This one's pretty common. And I love these steps because it's very specific. So this is based on antibodies not only reacting with the homologous antigens, but they can also act as antigens and react with anti-immunoglobulins. And I have a picture of this coming up next too. The antigen source to the specific antibody being tested is affixed to the surface of a microscope slide. Okay, so picture the nasal swab from the patient. Or you could just purchase them with them already being attached to a slide. The patient's serum is placed on the slide to cover the antigen source. If the antibody is present, it will bind to the antigen. After that, another step occurs where the fluorescent complex is placed on the slide and it would bind to the antibody-antigen complex. On the left-hand side, we have direct, and then on the right-hand side, we have the indirect immunofluorescent, where we had several layers of things that we're adding. We do have some new emerging labeling technologies coming out. The first one is quantum dots, or sometimes called Q dots. This is where semiconductor nanocrystals are used as a fluorescent labeling reagent. There's different sizes of crystals that produce different signals. The SQUID technology is a superconducting quantum interference device that actually uses magnetic particles, and the strength of the magnetic field would determine the amount of particles that were created. Luminescent oxygen channeling immunoassay, sometimes called LOCI. We see this quite a bit in some of our new chemistry analyzers, especially I think the Dade, the Siemens maybe analyzers are the ones we see this in. This is based on two different latex particles. We got a sensitizer particle and a chemiluminescent molecule. And when when they're close enough together, a single oxygen moves from the sensitizer particle to the chemiluminescent molecule, where it triggers that signal. And lastly, we have fluorescence in situ hybridization, or FISH. This uses a DNA technology where a specific nucleic acid sequence is bound and the chromosome is affixed to a glass slide. We almost call this gene painting, like painting. And I always think of those painted fish they had in fish tanks when I was a kid, where they actually were painted fish. So I always think of the gene painting with the fish in this one. So that concludes our immunoassay lecture for today.